Good day, grade 12 learners. Welcome to today's Life Sciences lesson. My name is Sina Sekheche, but I'm not alone today. I am with my co-presenter, Vedika Maharaj. And this lesson is brought to you by the Gauteng Department of Education in collaboration with Saibono Discovery Center. Right, so today we are starting a new topic, that is genetics and inheritance. Right, but for this topic, we are going to cover the first aspects of the topic, which are the monohybrid crosses, sex determination, and sex-linked inheritance. Now, let's get into it. So, we want to check what are the objectives that you need to achieve at the end of this lesson, or what is it that you shall know at the end of this lesson. So you must be able to know the concepts in inheritance. Now, genetics as a new topic to you, it comes with a lot of terminology that is new to you as a life sciences learner in grade 12, but you will see some terminology you will remember from grade 10 or grade 11. However, you need to learn these concepts in inheritance and how they work. Then you need to know the law of dominance. So there are laws that are governing genetics, or how we inherit genes, or how organisms, not only just us as humans, inherit genes. Then you need to understand those laws. So the first one is the law of dominance, and you will learn about the others. Then you must also know the format of representing a genetic cross. You want to check how do we put it, how do we write it from start to finish. So you must be able to know that by the end of this lesson. You must know Mendel's principle of segregation. So this will be your second law. So you must be able to know it and apply it. Then you must also know the types of dominance: complete, incomplete, and co-dominance. You need to understand how they all work, how the inheritance in these types of dominance works. And you must also know sex determination and sex-linked inheritance. I know some of these may be very vague to you as you are starting this lesson, but as we go on, you will be able to see what we are talking about. Now, let us move on. Before we move on, let us check what is it that you know, that you already know about a cell. It is going to build for this new topic that you are starting, which is genetics. Now, in a cell, you find this is a cell. So we can say it's an animal cell, and we can give our reasons as in grade 10. Then you find a nucleus. Now, inside this nucleus, you find a network. So let us write first the network. That network is called the chromatin network. You remember this from grade 10. And this chromatin network, like we have here, is going to untangle and form what we call chromosomes. You did this when you were doing, or when you were studying meiosis. Now you know that chromatin network is going to untangle, or is going to unwind and condenses to form a chromosome. So a chromosome is something that looks like this blue thing here. Then on a chromosome, you know this is where you find your DNA. Your DNA, you did DNA also in term 1. This was your first topic. You know that you find it on your chromosomes. Then on this DNA, a larger segment like this, a large segment of DNA that is coding for particular characteristic or a trait. A characteristic, we can call it a characteristic or a trait, is called a gene. Now this whole segment here is a segment on DNA that is coding for a particular characteristic. Characteristics may be height, eye color, hair, whether it's straight or it's curly, hair shape, or you can say that. Anything, all those characteristics are called traits, and we inherit them. So where do you get the information from? You get it from a gene. So now this brings us to what we'll be doing today. You must understand what is a gene. A gene is a segment of DNA. See this DNA here? This is a segment. It's not the entire DNA, only just a segment. So you can say here, when you try to maybe to be logical, you see just a small segment of that particular DNA is going to give us a gene that is coding for a particular characteristics. So our study now will be based on these genes, right? Going forward, we need to be able to say, what is it that you know about a chromosome? So you need to draw a diagram to illustrate a chromosome. So let us look at our chromosome, because this is where everything is happening. You remember that a chromosome is a diagram that has, you see, this is a full chromosome. It has one chromatid and another chromatid joined together by a centromere. And this is a whole chromosome. And on a chromosome, we have just now established that you find DNA, and on that segment of DNA, or a particular portion of that particular DNA, is called what? A gene. And these genes are what we are going to study. So this baseline assessment helps you to know or to get to understand where we are going with today's lesson. Right. Like I said to you, that there's a lot of terminology that you need to study for this section. You need to study. I'm not going to tell you the definitions, because the definitions are here on the presentation, and you also have them in your resources that you have with your teacher. But please be aware of what terminology that you need to study for this topic. The terminology include terms such as albinism, alleles. Now this one we're going to talk about a lot today, which are two alternative forms of a gene at the same locus. So you need to understand what is this. Don't worry, I'm going to break it down to you. Artificial selection or selective breeding, biotechnology, cloning, codominance. Codominance, we are also going to talk about it a lot today. So we will unpack it for you. Complete dominance, chromatin network, we've already spoken about it. A chromosome, we've already spoken about it. Dihybrid cross. A dihybrid cross, we're going to talk about it next time when we meet again, but you just know that this is where we are crossing two different characteristics. So we will do that at a later stage when you've studied monohybrid crosses. Dominant allele. Like I said, we're going to talk about the alleles. This dominant allele, what do we mean by that? Gene, we've already spoken about the gene. We said it is a segment of DNA or a chromosome that codes for a particular characteristics. And we explain the characteristics to be examples such as hair color, length, or whatever, eye color, you see, eye color. So those are characteristics that can be coded for by this segment of a gene or by this segment of a chromosome. Then gene mutation, you're also going to talk about that. A genotype, we're going to talk about that as well. A genome, this is a complete set of chromosomes in a cell of an organism. So the human genome, you will tell us how many chromosomes that it have. Then hemophilia, we're going to talk about that as well today. Heterozygous, homologous structures, homozygous, a hypothesis. This, remember, is a life sciences study, so you need to be able to hypothesize everywhere. Incomplete dominance. Today also we're going to talk about it. A locus. Let me explain a locus. This one I am so much interested in, because when I speak about it, I want you to be able to understand what a locus is. Now, a locus is the exact position or location of a gene on a chromosome. So remember, if we have a chromosome like this, right? And then we said this is a chromosome. And then here is a gene. So this area here, we call it a loci or a locus. So on the same location, or the position of a gene on a chromosome. So that position or the location, we call it a locus. So because the term locus is almost sounding like location, so you can remember locus is the position or the location of a gene on a chromosome, right? Then those are just terminologies that you need to be able to understand when we talk about this topic in general. But let us look at this father of genetics. I hope you know about this man called Gregor Mendel. This is Gregor Mendel, and he is regarded as the father of genetics because he is the one who studied the peas. He did his genetic studies with the peas, and he was too observant and he was curious to find out why do we have other peas that are wrinkled or others with yellows, that one with cotyledon or one cotyledon or two with cotyledon. So he studied the peas. Hence you see here, his picture is associated with peas. This is because he used peas to study genetics and to study inheritance. This is what helped him to come up with the laws of inheritance, to tell us that the dominant allele is going to do what, the recessive allele is going to do what, segregation, independent assortment. So he came up with all of those things while he was studying the peas or the inheritance of genes in the peas. So that study of his gave us a lot of information to what we know genetics to be today. Hence, he is known as the father of genetics. So when you are asked about the father of genetics, you must be able to say his name is Gregor Mendel. So whatever we are studying was based on his studies. Right. Now we've checked the terminology that we need to study. However, because this is deep jargon or deep terminology for life sciences, there are terms that may sound similar, and you find that you are using them interchangeably or you are confusing them. Terms such as chromatin versus a chromosome. Do you know the difference? And we can also put a chromatid also in the mix here. So sometimes you must be careful which one you are talking about here. So they are used interchangeably, but we are going to look deeper into these differences. Hence we have them versus here. So we look at each one. What is the difference between each concepts that are mentioned here? Genes and alleles. Sometimes even when we are teaching, you want to say a gene, you want to say an allele. So you must be specific which one you are talking about. So you can use them interchangeably when you actually mean the other. So we will look at what is actually a gene and what is an allele. Then a dominant allele and a recessive allele. So dominant and recessive. You must be able to put them into which one is dominant, which one is recessive, and they are also sometimes used interchangeably. Phenotype and genotype, you see, they sound almost the same. So sometimes when you want to say, when you are talking, you want to say genotype, you find yourself saying phenotype or phenotype, genotype, or when the question asks you, you don't know which one is which. And then homozygous and heterozygous. So these are some of the concepts that are used interchangeably, but let us look at each and every one of these concepts in detail now. The first one is chromatin versus chromosome. See here, chromatin versus chromosome. If you take a look at this cell over here on the left, on my left, this is a cell. It has a network. You see, it looks like a bowl with noodles. Now, those noodles, they represent what? A chromatin network. So you don't just say it's a chromatin, it's a network. So you see it is tangled, it's all over. But once it untangles, or we say it condenses, it will condense, meaning it will shorten to form what we call a chromosome, this here. So when this network here, these noodles in this bowl untangle or condense, they form a chromosome. And we know now a chromosome consists of one chromatid and another chromatid that are joined together by a centromere. So now you know the differences between a chromatin network and a chromosome. Now let us move to the next concept. Now this concept is where now sometimes the challenge comes. We're going to take a look at it slowly so we understand, because allele is now, we are bringing you a new terminology. But first, let us take a look at the gene. What is a gene? A gene is a segment of DNA. We loosely say that you have inherited your genes from your mother or your father. So what are we saying in essence? We are saying that you have inherited the DNA from your mother and your father. And with the previous topics, you know where you find the DNA. Now, on this DNA, there are segments that of genes, these segments that are coding for particular characteristics. An organism is made up of many characteristics. Many characteristics. You can take a look at yourself and look at your eye color, hair color, your height. Those are your characteristics. Where did you get those characteristics? From the genes that you inherited from your mother and your father. But what is a gene? Is that particular segment of a DNA or of a chromosome that is coding for a particular characteristics, meaning your characteristics. Let us look at the gene on this diagram that we have here on the right. So here on the right, you find that there is a locus. So these are chromosome, homologous pair of chromosomes. When we say homologous pair of chromosomes, remember we mean one comes from the mother. Let's say we are talking about humans. That will be the case, but in this case, remember, this is a flower. We are talking about the flower, so we'll talk about the male and female, right? So the homologous pair of chromosomes, here they are. On these homologous pairs of chromosomes, remember, for them to be homologous, we mean they have the same shape and the same size. You have inherited them during fertilization from the male and the female parents. Now, because they had the same shape and the same size, on the same location, you see here, on the same location, this is where you are finding a gene for flower color, meaning this is where the gene for flower color is going to be. Just check, when you talk about a gene, you don't talk about the characteristic itself that it has. You talk about the characteristics in the main, meaning the characteristic, not the allele. We're going to come to the allele. So the characteristic of this gene is the gene that is coding for flower color, but we have not mentioned which color. So you may find that on the mother's gene, the mother has an allele for purple flowers. What do we mean by that? An allele for purple flowers. What is an allele now? This is something new. We've said this is a gene because they are on the same location on these two homologous chromosomes and they are coding for flower color. But on these genes, we have alternate forms. What do we mean by alternate forms of a gene? We say these are alleles. Now, these alternative forms of a gene are localized on the same locus on homologous chromosomes. What do we mean by that? We mean on the gene of the mother, there is an allele for purple flowers. So the mother has her own characteristics. So the characteristic is found on the allele. Then the father also has his own allele for flower color, which is white. Now you see, they are on the same locus on the same location of these two particular genes. And they are alternative forms, meaning if you talk about height, height has alternative forms. You have tall and short. Then we will talk about another concept where we talk about the dominance and the recessive. This is where you will learn to say, how do you end up having the characteristic that you have? But on this is how now you define an allele. Moving on to the second concept, genotype and phenotype. What do we mean by genotype? Genotype, the name genotype, you see, has the genes. Can you see the genes with your naked eyes? No. Genotype is the genetic makeup of an organism, including all the DNA sequences that determine inherited traits. So whatever that is making you to be who you are today, we can't see with our naked eye, but we know that you have the DNA that has the genes that gave you the characteristics that you have. So those genetic makeup, that whole genetic makeup that makes you who you are, that we cannot see with our naked eye, is called a genotype of an organism. Then we come to the phenotype. The phenotype, pheno, physical. So you can say this is pheno goes with physical. Then something that is physical is something that you can see with your naked eye. What is the phenotype? Phenotype is the set of observable characteristics or traits of an organism. So these ones, when we say they are observable, it means you can see them with your naked eye, resulting from the interaction of its genotype with the environment. What does this statement say? It says, whatever that you see in the phenotype is influenced by what you have in your genotype, what we cannot see. The genotype is your genetic makeup that is inside, but your genetic makeup has the characteristics that makes us to see your physical attributes or your phenotype, meaning what we can see on the outside. I hope now you are able to understand the term genotype. When we're talking about the genotype of an organism, we are talking about the genetic makeup that is inside. When we are talking about the phenotype of an organism, we are talking about all sets of characteristics that are observable. And these characteristics are influenced by what? By the genotype. I think now everyone understands these two concepts. Moving on to the next concept, homozygous versus heterozygous. Now these ones are new. Homo, we learn in grade 10. Homo means the same. The same. Hetero will mean different in this case. So let us take a look at homo first. Homozygous. Remember, we've spoken about the alleles. We said the alleles are the ones, they are alternative or the alternate forms of a gene and for a particular characteristic. Meaning, you may find that there is short and tall. But let us use this example that we have here. Homozygous is a state where alleles of the same characteristics are both the same. Then the organism will be said to be homozygous. Take a look at this. These diagrams are diagrams that are representing diploid cell. What do you mean by diploid cell? It has a full set of chromosomes from maternal and paternal, meaning it has 46 chromosomes. Now, because this cell has a full set of chromosomes, if it was a human cell, that is just an example, then if human, then it will be 46. It's a full set from the mother and the father. Then in this homologous chromosomes, you see, homologous chromosomes, you are going to find on this low key, there is a gene that is coding for a particular characteristics. For this case, we can say eye color. Let's say eye color here. Eye color. This is a gene for eye color. Now, if it is a gene for eye color, you may find that the mother, let's say this one is coming from the mother, this one is coming from the father. The mother had an allele for eye color that is brown. Let's say this B is brown. Then we write it with the big letter B. Then the father also, you will find that by chance, he also, on this gene, he also has an allele for eye color that is brown. Now, this is our child. Remember, this is our offspring. Offspring, this is just to show you this is what they inherited already. So they inherited brown color from the mother. They inherited brown color from the father. Because they inherited those brown colors, and you find that those brown colors, let's say they were dominant, meaning they were visible. The mother has brown eyes, the father has brown eyes. We say they are dominant because they are visible. Now, because they are the same, for this child on this particular low key, these are the alleles for eye colors. They are the same. We say they are in a homozygous state because they are the same. But in this offspring, the second offspring, check, we are now going to heterozygous. If the alleles for the characteristics are different, so our heterozygous here will be different, the organism is described as heterozygous for that characteristics. Remember, we are only focusing on one characteristic at a time. Now, for this offspring, they have inherited, let's stick to our first example, from the mother, they've inherited what? Brown allele for eye color. When we talk about them in alternative forms, we are referring to them now as alleles, not as genes. Right, the brown allele for eye color. And then from the father, this offspring has inherited the blue allele for eye color. But this, because they are not the same, you see, there's a big letter B because we said brown is dominant, meaning brown is visible. So B, big letter B, small letter b. They are still talking about the genes, or they're still talking about the gene for the characteristics for eye color. But they are not the same. They are alternative forms. So, but because they are not the same, we say they are in a heterozygous state. So when they say an offspring is heterozygous, it means the alleles for that particular gene are not the same. And we will discuss later that when they are not the same, the dominant one is the one that is going to be expressed in the phenotype. Right. So, and then in this case, we also have a homozygous. But this homozygous is homozygous recessive. Why do we say it's recessive? Because all of them are small, bringing us to the next term that we need to discuss. Dominant versus recessive. When we say something is dominant over the other, sometimes at the back of our mind, we think of something that overpowers. So say overshadows, it's dominant. But let us take a look at the scientific or the biological concept that we are discussing here. We say an allele that masks, so it masks, it will cover. You see, if something is dominant, it will come on top. So it is going to mask or suppress, you see, suppress the expression of the allele partner. Now, I like this allele partner story because when you talk about allele partners, yeah, alleles work in partners. They will always work in partners because an organism will have both alleles, one from the mother, one from the father on the same gene. So, in that partnership of alleles, there will be an allele that is dominant over the other. So the one that is dominant, it is the one that is going to be shown or that is going to be expressed in the physical attributes of that particular organism, the one that you're going to be able to observe. Right, let us take a look at this diagram here. You see this P here, this is a P, it is green in color. But when we look at the genotype of this, what made it to be green, you go and look at what is this? The allele. So these are alleles. Why do we say they are alleles? Remember, they are on the same loci. You go and look at the allele of the mother or the parent one. Let's say these are just parent 2, because we are talking about P's. Parent 1 and parent 2. Look, parent 2 has round dominant, meaning it is round, or we are talking about the shape. So the shape is the characteristic here. So this one is round, this one is wrinkled, recessive. Because round is dominant, when you cross, or this organism consists of these two, the phenotype is going to be round. Why is it round? Because this R for round is dominant over the small r in this partnership of alleles. In the partnerships of parent 1 and parent 2, the alleles came to say parent 1 gave a recessive wrinkled allele, parent 2 gave a round dominant allele. So in this partnership, in this offspring, what is going to be dominant is this one of round, because the round, we are told that it is dominant over the recessive that is wrinkled. Therefore, our offspring, that's how they ended with the shape of a round, or of it being a round P. Then, a dominant allele can be expressed in a homozygous state. Remember, we've just spoken about homozygous. It can be when there are homozygous alleles, or alleles that are the same. Dominant, it can be expressed like this, or it can be expressed in a heterozygous state. Also, this will be dominant. T, big letter T and small letter t. So it can be expressed in those two ways. Let us look at the second term, which is recessive. Recessive, remember, is the one that is being suppressed. Now, let us take a look at our phenotype for P number 2. It is wrinkled. You see, this is wrinkled. Why is it wrinkled? Let us check. And a recessive allele is an allele that is suppressed when the allele partner is dominant. Remember, we said when the allele partner is dominant, then recessive. So the recessive is the one that is going to be suppressed. It's going to be masked. The recessive allele or the recessive trait will only be expressed or seen in the phenotype if both allele for the trait are homozygous. Please, this one, you need to check. Let us take a look at our parent 1 and parent 2 when they donated. So remember, these are not parents, they just gave to this offspring. So what they gave is that parent 1 gave a recessive wrinkled. Parent 2 also gave recessive wrinkled for what? For the shape of the P. Now, this shape, because it is recessive plus recessive, it can therefore give us the phenotype of wrinkled. But if it was in this way, if it was written in this way, we know because there will be a letter big R which will be dominant over the small r. This case, we cannot have a dominant, we cannot have a P that is wrinkled. This P is going to be round. Why? Because big letter R is dominant, round is dominant over recessive R. Therefore, you are not going to have a situation where the recessive is expressed in a heterozygous state. It can only be expressed in the phenotype if it is in a homozygous state. So for it to show in the phenotype, it must be small letters together, because if there's a big letter there, you know that big letter is going to be dominant over the recessive one. So that is the recessive and the dominant story. Then we come to the Mendel's laws of inheritance. The law of dominance says that when two homozygous organisms with contrasting characteristics are crossed, all the individuals of the F1 generation will display the dominant trait. What does this mean? This is quite a lot of English here. Two homozygous organisms, meaning when they say homozygous, they must be the same letters. So the letters must be the same. When you cross homozygous, okay, homozygous dominant, let's say homozygous with homozygous. Remember, with genetics, we are crossing. We want to play with them. We want to see what we are finding. they say all the individuals of the F1 will display the dominant characteristics. So if you have all dominant, all the offspring or the filial one generation, these are your offspring, are going to have that dominant characteristic. So let us take a look at this diagram here. You see there's a yellow P that is male. It has a dominant allele for color yellow. Then you have the recessive green. So when you cross this yellow with this green, in their homozygous state, let's say this green is small y's and this is big y's, all your offsprings are going to be yellow. Why yellow? Because all this big Y will be in all of the characteristics crossed. So this is what is meant by Mendel's first law of inheritance. Then an individual that is heterozygous for a particular characteristics will have a dominant trait as a phenotype. What does it mean? What does this mean? If you have an individual with big Y and small y, this big Y will always tell us what is that characteristics in the phenotype, meaning the dominant one is the one that is going to be shown in the phenotype. So that is what is meant by this second statement. So you will see these laws will make sense to you as you start applying them. The second law is the Mendel's principle of segregation. What does this mean? An organism possesses two factors which separate or segregate so that each gamete contains only one of these factors. Now, let us take a look at these chromosomes that we have here. We have 3 pairs of chromosomes, the orange ones, the blue ones, and the green ones. During meiosis, these are going to separate. What does this mean? Each gamete, remember, meiosis is the process where we are forming gametes. So each gamete is going to have one of each of these during that segregation. So let us take a look at what is happening inside each cell. First cell, you have orange, green, and blue. Orange, green, and blue represented. Second cell, orange, green, blue. Third, green, orange, blue. Fourth, orange, blue, green. So all of them, they have segregated. And when they segregate, you see, they are separate. So it means that an organism is going to possess 2 factors which are going to separate so that each gamete contains only one of these factors. So this is where you are going to separate your alleles, because those alleles are coming with the chromosomes. You see these chromosomes here, they have alleles. Now all of these alleles have separated during the process of meiosis, and this is what also brings about genetic variation, because then they just separate like that. Then Mendel's principle of independent assortment. Various factors controlling the different characteristics are separate entities. Now, separate entities. Independent assortment. What makes the color of your eye will not influence what makes the shape of your eye. So you can inherit the color of your eye from your mom and the shape of your eye from your dad. So it does not mean that if you get one characteristic, you're going to get all of them from only the individual parent. So it means they are separate entities. You can take a look at this example that we have here. You see this offspring here? Offspring has inherited the brown eyes which were dominant in this parent, and also, but not the oval shape of the eye. Then the oval shape of the eye, they inherited the oval shape of the eye from this second parent who had blue eyes, but that were recessive. But the shape of the eyes were dominant. Let's say this big eye, they talk about the shape. These ones, they talk about the eye color. So from this one, the shape was recessive. It gave us oval. But this one, the parent with the blue eye had round eyes, which were big, and it was dominant. So during this inheritance, the factors controlling these different characteristics are separate entities, not influencing each other in any way, and they are sorting themselves independently during gamete formation. So you will not say you inherit all from one. This is what this principle actually is talking about, right? Then we have examples such as the monohybrid crosses. This is the first thing that we are going to do. We've been talking about terminology. It's best with genetics to see terminology in action, because if you don't apply it, if you don't read questions on it, there's no way you'll be able to apply it. So how are you going to apply it? First thing now, we are going to do genetic crosses. You want to find out how a particular individual inherited a particular characteristic. So we are going to start with the simplest one, which are the monohybrid crosses. What do we mean by monohybrid crosses? These are a cross between two organisms, but involving only one trait. Only one trait. There are no other characteristics, so you only focus on one characteristic at a time. An example can be here. If you cross, we are going to play now with scenarios. In genetics, you play with scenarios, you listen to the information that is given, and then you interpret. Then here we are crossing a true breeding pot plant. When we say true breeding, please just know that they say it is homozygous. Ne? Homozygous. And you know homozygous means what? It has, the alleles are the same. Right. Then with a true breeding, green. So we are crossing a green pea. We are now Mendel. Green pot with yellow pot with a green pot. So because they said they are true breeding, it means they are homozygous here. This is what they mean by true breeding. Hence, I say you need to be able to read the questions nicely. So then we know that green, in this case, green is dominant. Very important. You need to establish which one is dominant, which one is recessive. So yellow is recessive. Right. Because it is recessive, and when you use a letter, you don't use different letters. You don't say G here, and then this side you come and use a different letter to say yellow, Y. You're going to use an alternative form of the letter you first chose. So if G is going to be the letter that you decided, the dominant will be big letter G, and the recessive will be small letter G. But we know you are talking about the P color, which is our trait in this case. So let us cross it and check how do you do a monohybrid cross. Simple, use a Punnett square. Let us check. You are going to cross this G with this G, and then you get this big G, small g. Remember what the principle said. The law of dominance of Mendel. He said that if an organism is in a heterozygous state, the big G is going to be the one that is shown in the phenotype. Hence, because we have big G here and small g, hence you find a green pot here because of this big G. Then you cross this big G with this small g, you get big G, small g. I think now you understand the math. This one with that one still gives you the same, and this one and that one still gives you the same. So what can you say? When you cross a true breeding or a dominant green pea with all the recessive, all the offspring are going to be green. So I will show you the situation of what just happened here. All green. So this is just simple. You see, what you need to know is whether you have alleles that are homozygous or heterozygous, which one is dominant, which one is recessive. Once you've identified that, let me tell you, you'll be able to tell us your cross and be able to tell us a story about the offspring. All of these that are in here represents the offspring. So that is just an example, but we want to go further now. How do you write a genetic cross for you to get marks? that was just a way to show you how the crosses work, but there is a particular format that you need to follow for you to collect marks. It must tell us a whole story of what is happening. So you will get a scenario telling you that when you cross a parent, blah, 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 this is what is going to happen. Then once you get that story, find the visible trait or the phenotype. Example, when they say when you cross a tall tree with a short tree, already those are visible things. Those are the phenotype. Then what are you going to do? You're going to write it here. You are crossing a tall tree with a short tree. You write it as full as it is, the way it is explained in the question. When they say hemophilic mother with a hemolymph, write it as that, because those are physical attributes. So this is the first thing that you do. You write P1. And with the P1, these are your parent generation, your first parent. Then you write their phenotype. So the question will always tell you how the parents are, because it wants you to find out how the offsprings will be. So the first thing, you are going to write the phenotype of those parents, and you get a mark for that. This is a free mark. Then you come with the second thing, the genetic makeup now of those parents. This is where you're going to listen to the question where they say, the parent was homozygous for tallness or was heterozygous for shortness or what? No, it cannot happen heterozygous for shortness. Remember what we said, recessive can only occur when they are in a homozygous state. Now, you must listen to the question and underline. Did they say the tall tree is dominant? Is it homozygous? When they say it's homozygous, you know you are going to represent it with the same letters. So you will say tall, and then short, homozygous, it will always be recessive. When they say it's recessive, you know it is small letters, both of them. It cannot be in a heterozygous state. Remember that. This is the stage where most of you will make mistakes, but please be careful of the letters, what you are using there. Then this is what we call the genotype of the parents. So all this information you got from the question or the scenario you were given. What must you do now with this information? You will say, now you are applying the law of segregation. During meiosis, you write meiosis. You see, meiosis comes before you write gametes. Remember, meiosis is a process that is giving us the gametes. You are forming the gametes. And we said now the law of segregation is that all of these are going to segregate so that each gamete contains one. So what must you do? You are going to separate them into each gamete. How do you separate them? You say one gamete will have big T and another gamete will have big T. So it's best to write them, to put circles around them, because sometimes when you put commas or you put spaces, we can't see that you have separated the gametes, or the gametes are still close. And when they are close to one another, they are called alleles. But when they are separate to one another, we know that they are gametes because they have separated. So use circles, right? Then you can also separate these ones, T and T. That is what you do. And then here, from the gametes, these gametes during fertilization, remember random fertilization, you write meiosis, and meiosis gives us gametes, and these gametes are going to fertilize. Now, during this process of fertilization, this is where random fertilization is happening. You do the crosses. You see, this means you multiply this one with this one, this one also with the second one with the first one, second one with the second one. You see, this is what is meant by all of these crosses here. Then you are going to get your results. What do you get as results? Because it's so easy here. You get big T when you get this one with small t, big T with small t, big T with small t, big T with small t. So you see now you have given us the genotype. But whose genotype is this? Is the genotype of the F1. You write F1 here. What is F1? F1 is now your offspring, your first filial generation. So it is your offspring. With the offspring, you must tell us first what is their genetic makeup, because the genetic makeup comes from this fertilization. Then you write You write now the phenotype from the genotype. Remember, the genotype gives you the phenotype. You write now these are all tall. Remember, tall, tall, tall. Why tall, tall, tall? Because there's a big T which is dominant over short. Then you are going to tell us that all offspring are tall. And sometimes they may ask you for a percentage, or sometimes they may ask you for a ratio. So you must read your question carefully. So where are you going to get your other marks? Look, for writing P1 and F1 here, this one and this one, you get a mark at the correct place. Then for writing meiosis and fertilization, meiosis gives us gamete, then those gametes are going to fertilize. That in that order. So you're going to write meiosis, fertilization, then you get another mark. This P1 and F1 and meiosis and fertilization and the phenotype of the offspring, I always say those are the 3 marks. Please make sure that you collect those marks. Right, this is just another way that you can use is when you don't want to do the crosses, especially when you have a dihybrid cross, you can use a Punnett square. A Punnett square helps you to see actually where your gametes are, and then you will write your offsprings here, and then it will give you. So this is just the same as that one, but here you don't use that cross method, you use a Punnett square. And remember those 2 marks for P1 and F1 and then meiosis and fertilization. These are 3 marks that you need to remember. Now, in inheritance, there are 3 types of scenarios that you can have. You can have complete dominance, incomplete dominance, and co-dominance. Right. Complete dominance, I want to explain to you, what do we mean by complete dominance? Complete dominance. Try to break out your words before you even try to attempt. Complete dominance means that this thing is completely dominant over the other. An example that we have here is, this is now, okay, let us look at the explanation first. This is a genetic interaction where one allele of a gene suppresses the expression of an alternate allele in the F1. So you will always have in a heterozygous state, the one that is dominant or suppressing the recessive one. We call that complete dominant. So it is completely dominant over the recessive one because it is the one that is going to be shown in the physical attribute or in the phenotype of the offspring. Let us take a look at this example that we have with the flowers. We don't have the white flower here. Look. When you cross a red flower, where red is dominant, you see, red is dominant. How do I know this? Because I see big letter R. You cross the red flower with a white flower. You cross a red flower with a white flower. But all offspring are red. What does this tell you? It tells you that the color red is completely dominant over white. What do we mean by that? White can only be expressed in a homozygous recessive. Then all the offspring will have big R, remember one from each parent. Big R, small r, big R, small r, big R, small r. But this big R, because it is in all the offspring, it means all offspring will have red color, meaning all the flowers here will be red. That is complete dominant. We can say now that red is completely dominant over this. This is where you are going to listen to your question where you are given such. Then an example of complete dominance is that there are 4 types of causes that you can have in a complete dominant story. Is that you can find that a homozygous dominant, a homozygous recessive, meaning heterozygous and heterozygous will be with, so homozygous dominant versus heterozygous. So you will also have this with this. So you see, these are you need to be able to understand what they mean by what? Dominant, heterozygous, recessive, heterozygous, homozygous. So those are the three words that are playing here. Homozygous means the same with heterozygous. You see? Oh, homozygous recessive. So here is small letter H. Right. In the rabbits, the allele for black color, B, is dominant over the allele for fair color. Letter B. The inheritance of fair color is controlled by two alleles, namely black fair and white fair. So we see now white is recessive, black is dominant. So the first scenario where you have homozygous plus homozygous dominant plus homozygous recessive, all offspring are going to be 100% black. Why? Because the big letter B is dominant over this. So this one is the example like the flowers that we did. So this is the first example. Then the second example where there is heterozygous with heterozygous. Look, heterozygous with heterozygous. Offsprings 75% are going to be black, not all, because there is this one, 25% is going to be white. So the ratio will always be 3:1. So you need to learn that when you cross a heterozygous with a heterozygous, if you don't get 3:1, there's something that went wrong in your cross. So this is something that you need to know of by heart or at the back of your mind. Then if you have a situation with a homozygous dominant and something that is heterozygous, so let's look. Homozygous dominant for black color with heterozygous. What are the chances? The offsprings are going to be all 100% black. So you check your statement with what you get at the end so that you can verify your cross if it is correct. Then another situation that can occur in complete dominance is homozygous recessive. Here, homozygous, same, recessive. When you cross it with the heterozygous, the offsprings are going to be 50-50. 50% are going to be black, 50% are going to be white. That is how you compare. That is a situation. I would like for you to do this activity with your teacher because we don't have much time for this activity. This is quite what is this? Intense topic. Right. Then another type of dominance, we have two types again. We have three types. So we've spoken about the complete dominance. Now we are talking about the incomplete dominance. Check here. When you are crossing our red flower, which is dominant, with a white flower, this white flower in this case also is dominant. What is going to happen? Our offspring is going to be pink. Why is it pink? Neither one of the two alleles of a gene is dominant over the other. So in incomplete dominant, neither one is dominant over the other. So what happens? They are going to form now an intermediate phenotype, meaning the phenotype that is different from both the parent plants. So it is going to be the intermediate one, the one that is different from what you had with the parents. Then you know that type of cross is incomplete dominance. And you must also check the letters here. We write them in capital letters for both because both of them are dominant, but none of them is dominant over the other. Therefore, they are expressed in an intermediate phenotype in the offspring. Then another type of dominance is the codominance. Now, what is codominance? Let us take a look at the example that we have here. If you cross a red flower, look at the letters also. Homozygous dominant. With a white flower, also homozygous dominant. Meaning white is dominant in its own way. Red is also dominant in its own way. Then you cross those. What are you going to get? The offspring will have those colors or of both genes are equally dominant and they are express themselves in the phenotype in a heterozygous condition. Now look, in a heterozygous condition, you're going to have this R and this W. Then the phenotype of this offspring is going to have both the colors mixed. You see now you have a red and a white flower, not only just a pink in this case. When it's pink, it's different from the parents, we say it is incomplete. But when it is both of them are equally expressed, it is codominance. So they found a way to say, let us both be expressed in the phenotype of the offspring equally, because none of them are also dominant over the other. So that is another situation that you can get with codominance. So their crosses will always have this. But with codominance and incomplete dominance, where are you going to get the information? Because the letters are all in, remember, look at this example. You see, this is incomplete dominance, and this one also is codominance. How do you know that which one is which? Because we all use big letters here. You must listen to the statement that you are given in the question. When they tell you that you cross a white with black, the offspring are gray, what is that? It is an intermediate color. Then you will know that it is an incomplete dominance. But when they say that you cross a red and a white cow, and then you get offsprings of all the cows are roan. What is that? Did we speak about the roan with the parents? No, that is now the intermediate also. But when that is also a codominance, it means now it combined the two in the offspring. So look at your questions and be able to answer those questions based on this diagrams. So also this activity, please do it for homework to make sure that you understand how you need to go about solving the genetic crosses. I would have loved to do these activities with you. However, we are running short of time. Please make sure that you complete these activities. Then we'll mark these activities, and your teacher will mark these activities with you to make sure that you understand. So for today, my story will end there, and I will leave the presentation to my co-presenter Vedika Maharaj to take you through the sex-linked and sex determination, sex-linked inheritance and sex determination. Thank you.
Welcome back, grade 12 learners. Thank you, Sina, for that wonderful presentation and taking us through the introduction and the monohybrid crosses, the incomplete, complete, and codominance. And I want you to not forget about those when we are moving on now to the gonosomes and the sex-linked inheritance. Okay, do not forget about that. We are still going to use that information, and all the terms that she taught you, we are still going to use it in the rest of the lesson. Now I want to bring you to something to remind you again about going back to our human somatic cells and our gametes. Okay, so if we remember a somatic cell, the nucleus of a somatic cell, it has chromosomes, right? Do you remember how many chromosomes there are in a somatic cell's nucleus? There are 46 chromosomes, right? Which essentially are 23 pairs, right? 46 chromosomes, which are essentially 23 pairs. And 44 of those 46, which are 22 pairs, are autosomes. And 2 chromosomes out of that 46, which is the 23rd pair, and those are called gonosomes, also known as sex chromosomes. Okay, sex chromosomes is also accepted. However, gonosomes is the proper life sciences term, and that is the biological term that we do prefer you to use. Okay, so as I said, we have 46 in total. We have 44 autosomes and we have 2 gonosomes. Okay, those autosomes, those 44 autosomes are just our normal chromosomes with their normal shapes and sizes, whereas our 2 gonosomes they have a particular shape and size. Okay, they are distinguished by a large X chromosome and a small Y chromosome. So if an individual is a male, then in their somatic cell, in the nucleus of the somatic cell, if it's a male, the gamete will have, the gonosome will have a 1X, and the other gonosome will be a Y. So then it will be XY, and that is at pair 23, as I said, right? But in females, the 2 gonosomes are 2 identical X chromosomes, right? And that is depicted as this, and it is at the 23rd pair. Okay, so if you look at the picture here that I have on the right hand side, you can see the big X chromosome and you can see the smaller Y chromosome. Okay, so now let's just talk about all of what I've said and put it all together. Somatic cells of a male will have 44 autosomes plus an X and a Y. That XY are the gonosomes. And a gamete of this one will now have 22 plus X or 22 plus Y. Why am I saying this? Because remember, when we have our somatic cells, we are going to eventually, in our somatic cells, those are our body cells, right? But in our gamete, remember our gamete has half the number of cells. Our somatic cells are diploid. And our gametes are haploid. So essentially what we do is we need to halve this autosome number, which was from 44 to 22. And if you're going to halve this X and Y, then we are going to have a gamete that can either have X's or gametes that can have Y's. Okay, you understand that? But in a female, we also have the situation, but because females only have 2 X gonosomes, their gamete is going to have 22 plus X. So all the ova can only have 22 plus X, whereas sperm cells can have 22 plus X or 22 plus Y. Right, now we move on to sex determination. So now you basically figured out, as I told you, that we have autosomes and we have gonosomes, and the gonosomes are the ones that are going to determine the gender of an individual. The gonosomes determine the gender of an individual. So here I have 2 karyotypes, okay? And there's a female and a male. So the first 22 pairs, which I said to you, which are 46 chromosomes, right? those are autosomes. And the last pair, which is the 23rd one, are the gonosomes. And it is XX in the female and XY in the male. So that's how we distinguish. We look at the 23rd pair and we see, what do the gonosomes look like? If there are two identical X chromosomes, then it's a female. And if it's an X and a Y, then it's a male. Can you see how all of them are in pairs here? They are in homologous pairs. Right, so what we have to look at over here is that if you have to be asked to solve, this is how we can use a genetic cross or a monohybrid cross to basically solve our sex determination and our ratio of how many males to females. What is the probability or the chance of how many females or males a couple can actually have, right? So let's go through the template again that Sina has given you with the P1 and the F1, the meiosis and the fertilization. Okay, so you just plug it in over there. What is the phenotype we are looking at? We are looking at a male and a female. What are the genotypes of that male and female? So we look at the gonosome. So the male is going to have XY and the female XX. The gametes, law of segregation, you apply that, you're going to have X and Y and X and X. Plug it into our Punnett square, and eventually you will see there the genotypes that we get. We get 2XX and we get 2XY, which means there are 2 females and 2 males. So if you are looking at percentage, it's 50% male, 50% female. So there's a 50% chance that you can have a male and a 50% chance that you can have a female. That is with every male and every female couple that are going to have children. There's a 50% chance that you can have a boy and a 50% chance that they can have a girl. And the phenotypic ratio for this would be 1:1. So during meiosis, males produce, I explained this one to you already, what happens during meiosis about the X, 22X and the 22Y. Okay, depending on which sperm reaches the ovum, there is a 50% chance of the zygote being male and a 50% chance of the zygote being female. So what we mean by that, remember that the ovum can only have an X, but the sperm can have an X and a Y. So it's actually the sperm that's going to determine whether the zygote is going to be male or female, because that sperm can either have an X or a Y. If that sperm has an X, and that sperm is going to fuse, that that nucleus is going to fuse with an ovum, that's another X. Then we're going to have 2X, and that means that that zygote is going to be female. But if the sperm has a Y chromosome, then when it fuses with that ovum, we are now going to have XY. Oops, sorry. Right. So here we have Lindiwe has 2 sons and she is now pregnant for the 3rd time. Use a genetic cross to show the percentage chance that this child could be a boy. Okay, so now basically it's exactly what I did previously. You just do it again, and this is how you're going to prove. And then you need to answer the question. They're asking us what percentage chance that this child could be a boy. So we look there, and as you know, you're going to get 50-50, but they wanted you to work it out. You had to show a genetic cross. So you could not just say 50% females, even though you know that. The teacher has taught you that, right? But they want you to do a genetic cross, and then you will see that it is 50% males. Right, let's look at this activity. The diagram below represents the chromosomes from a human somatic cells of 2 individuals who are twins. Define the term somatic cells. So somatic cells are your body cells, or you can say they are all the cells, all the cells of the body except for the sex cells. Sex cells are gametes, right? Name the specific type of chromosomes numbered 1 to 22. So I just taught you that. All of them are called autosomes. Each of the pairs shown in a homologous pair of chromosomes. Each of the pairs shown is in a homologous pair of chromosomes. State the origin of each chromosome in a homologous pair during zygote formation. So they're saying that during zygote formation, these ones are going to pair. Right? Because remember, it will be from the gamete will come one, from another gamete will come from the other. So what essentially are we looking at here? We are looking at the fact that one chromosome is going to come from the mother and one from the father. Explain one observable reason why two individuals are not identical twins. So why are these two not identical twins? So if you have to look at all of their chromosomes, all of them look pretty identical, right? But let's look at number 23. That is the first giveaway. Identical twins have to be the same gender. And in this instance, one is a boy and one is a girl, and therefore they cannot be identical twins. They can still be twins, okay? They will be then fraternal twins, which means that they are not identical. Right, now we're going to look at sex-linked inheritance. Gonosomes carry sex-linked genes. So all of the crosses, the monohybrid crosses and the inheritance that Sina has done from the beginning of this lesson, all of those genes are carried on autosomes. Okay? Well, in a human, if the human ones that she did, okay? They are all carried on the autosome. Okay? So human, if you're looking at a human, all the traits of a human, all the traits that a human would have, all of those genes of those humans would be on autosomes. But there are types of inheritance that occur on the gonosomes. These are called sex-linked inheritance. The allele for the gene is carried on the X chromosome. Very important for you to understand, grade 12, is that the allele for that gene is carried on the X chromosome, not on the Y chromosome, on the X chromosome, the X gonosome, basically. And when you are doing questions on this, the question will indicate to you whether the trait is sex-linked or not. The question will tell you, okay? So if we have to let's look at the different situations that we can get. So firstly, you are going to mention whether the person is a male or a female, okay? That is part of the phenotype when you are doing sex-linked inheritance, okay? So in a normal female that is homozygous, okay? We are looking at if it's so when we are looking at the sex-linked also inheritance, a lot of the time we talk about how the person is affected or not affected by the characteristic, okay? But you must please not use these words, especially not normal, unless it is stated to you in the question. We'll look a bit more about that later. But I just want you to look for now. If it's a normal female, okay, and it's the possible genotypes for that female, it would be NN. So the female will be the X's, and the allele that is carried on that X is depicted with a superscript. Okay, so it's going to have a capital N superscript, and then that is going to be homozygous. Then another normal can also be in the heterozygous condition, because remember, this is the dominant. Okay, if we don't just use the word normal, we can just say, okay, we have the normal inheritance, right? And then it will be, we're using normal here because it's just showing you the generic examples, okay? But when we do specific examples, you will see that we're not going to use the word normal, right? Then we come to an affected female. So this means that this person has two recessive alleles, because remember what Sina said? The dominant is going to mask the recessive. That's why in this situation, we are saying that the person is still normal because the capital N is masking the small n, right? The affected female is homozygous. So now there's two recessive alleles. So the two small n's. So if there are two, that means that that recessive trait is going to be in the phenotype, depicted in the phenotype. Now we look at the males. The male is a little bit different. As you can see, because the Y chromosome does not carry the sex-linked inheritance, it's only the X chromosome. Males only have one X chromosome. So in order for the male to have that phenotype, he will only need to have one of the alleles on the X chromosome. So in this instance, he only has one X with an N, capital N. Therefore, in this phenotype, this individual is going to be normal. But if he inherited the small n, which is the recessive allele, then also in his phenotype, it will be depicted as the recessive trait. Okay, the recessive trait will be in the phenotype in a male, whereas in a female, the female would need two of those to be able to be in the phenotype. This is a generic approach to sex-linked phenotypes and genotypes. Okay, so as I said, this is a generic approach just to teach you the differences and how they can be depicted. Now we're going to do specific examples that you need to know. Right, so you need to know about hemophilia and you need to know about color blindness. It's part of your examination guideline. So if they ask you to do crosses or any information on hemophilia and color blindness, firstly, you need to know that they are both sex-linked inheritance. And it so happens that both of these are disorders. Okay, most of the time, the sex-linked traits or genes that are carried on the sex-linked has got to do with disorders, right? So let's look at what hemophilia is. Hemophilia is you need to know this. Hemophilia is the inability of the blood to clot due to the blood clotting factor. If the person were to cut themselves, the wound would continue to bleed. So usually when you cut yourself, a scab will start forming, the blood will start to clot, okay, and will form like a closure so that you stop bleeding. But now when somebody has hemophilia, they don't have that blood clotting factor, so they will just continue to bleed out. Okay, but now let's look at the genetics behind this. If we've got a father without hemophilia and we've got a mother without hemophilia, both of these individuals do not have hemophilia. However, what do you know about genotypes that Sina has taught you? That every genotype has 2 alleles. So for every trait, there are 2 alleles. So in this instance, this father has X, okay, and his X is now, we can assume now that it is going to have, he's got a capital H, right? Because now he's fine. Whereas the female is going to have a capital H and is going to have a small letter H because this individual is not hemophiliac. Okay, so but this individual, as you can see from here, it still is a carrier of the gene, which means that part of their genotype, one of the alleles is the recessive allele. So they can pass that recessive allele or onto their offspring. So what happened in this instance is that, can happen is that they are going to have they can have an affected son, right? A son with hemophilia. Why? Because this son is going to get this affected allele from the mother. And then for it to be a boy, how do we know that this X is coming from the mother? Because a boy has an X and a Y, a male, right? So that X has to come from the female. But then to make it from the mother, but to make this individual a male, the other allele had to come from the father. Okay, so that's how we get to the X and the Y. Why do we have an X and an X here for the female? Because the father gave an X and the mother gave an X. So what is the possibility? Number one, the son can definitely have hemophilia. But there can also be a son that does not have hemophilia. Why do we have an unaffected son? Because the mother also has a normal allele that could go to the son, and the Y will still come from the father. Right. Why do we have a normal daughter? Because there's a normal X coming from the father and a normal X coming from the mother. Why do we have a daughter that can be unaffected but be a carrier? Because she can get that recessive allele from the mother and then the normal allele from the father. So essentially what we can see here is that the father can only pass the affected allele to his daughters. If the father has an affected allele, so if the father had a problem, he can pass that on to his daughters, not to his sons, because he's going to give a Y chromosome to his sons, but he can give the affected allele to his daughters. But a mother can pass the affected allele to her sons and her daughters. So can you imagine that? She doesn't even have to have hemophilia, but because she's got the gene, she can pass that allele to the son or to the daughter. Color blindness works exactly the same way, so I'm not going to go into the detail of that, but I want to explain to you what color blindness is. This is when a person who is colorblind is unable to tell the different colors apart. For example, with red-green color blindness, it's caused by an absence of proteins that make up the red or green cones of the retina of the eye, resulting in the person not being able to tell the difference between red and green. So if we look here at the bottom, this is normal vision. You can see this is green, this is yellow, and this is red. But when a person has red-green confusion color blindness, you can see that everything looks like yellowish. We've got like a mustard color going on. Can you see how this green is looking for this person? Yellow would look yellow, and then this red would also look like a bit of a yellow. So that is how they would see. But the cross works exactly the same as hemophilia, and the same rules applies, that a male that is the same things that we said is that a father, let's just remind ourselves what we said over there on the previous one. The father can only pass the affected allele to his daughters, and the mother can pass it to both. Right, here we're going to have a look at a cross. Okay, it's already up here, but this is an activity, but it's an activity that we do together. Okay. Brown enamel of teeth is a sex-linked trait. A dominant allele on the X chromosome causes brown teeth in humans. Okay, so that's actually a thing, right? And it's carried on a it's carried on a X chromosome. And it is by a dominant allele. It's very important, grade 12, that when we get situations like this, we underline everything, okay? And we really engage with it. But let's read it first and I'll teach you how to do it. Brown enamel of the teeth is a sex-linked trait. A dominant allele on the X chromosome causes brown teeth in humans. A man with brown teeth married a woman with white teeth. Use a genetic cross to show the possible phenotypic ratios of their children. Use X superscript B for brown teeth and X superscript small letter b for white teeth. So I want to remind you again, the allele for the trait is carried on the X chromosome. The allele for the trait. Okay, so when we are talking about this, then we are talking about the allele for B, capital B. The allele is carried on the X chromosome. Okay, the allele is not B, it's X, and the allele. What I want to explain is that what learners are doing. Okay, we'll look at it when we look at the diagnostic. Let's first do the activity. Okay, brown enamel of the teeth is a sex-linked trait. So we're going to tell you brown enamel, and they're going to talk about white enamel. So here we have brown teeth is married to a woman with white teeth. That is our phenotype. When you are looking at the question, it's very important that we do that this is the phenotype. What is the dominant allele? This one is the dominant allele, okay? Because it's telling us now that it's the dominant trait is the brown one, okay? And then this one will be the recessive one, so this one is for white. So you need to put the phenotype and you need to talk about the genotypes when you are doing this. What do they want us to know? The phenotypic ratio of the children. So that is what you need to understand. So when you are answering the question, you know that the compulsory mark is going to be for the phenotypic ratios. Right, so here we go to P1, phenotype. Where do you get the phenotype, grade 12? You get it from the information given to you. They told you it was a male with brown teeth and a female with white teeth. You write it in. What are the genotypes? Immediately, because you know it is sex-linked, because you saw the alleles over here that was given to you, they are showing you here that it's X superscript B, right? Okay. This is how it is written, and then we have another one over here, focus 2 recessive for the white teeth. Okay, so the genotype here for the male, it's a capital B because he has the brown teeth. So then we have meiosis happening. We get the gametes. Okay, you separate them, you get the gametes, you plug the gametes into your Punnett square, and what do we have here? So look at the genotypes we have here. Can you see this is heterozygous, and here we have 2 recessive alleles with the male. So what do we have? We have one, we actually have 2 girls with brown teeth, and we have 1 boy with white teeth. So can you see, even though the father has the brown teeth, he actually, he cannot give that allele to his sons. He only gave the allele to his daughters, and he gave it to both of his daughters. So we have 2 with brown teeth and we have 2 with white teeth. But the phenotypic ratio must always be written in its simplest form. Right? Simplest form. And that's why we have 1:1. Right, brown enamel of the teeth is, okay, this is the same question, but the question itself is explain why more males than females have white teeth. Okay, why are there more males that actually have it? And the reason why is because I've been explaining it all the time. Males only need 1 X chromosome. The chromosome does not have, the Y chromosome does not have an allele, right? So males only need to have the 1 X chromosome. And they have to inherit only 1 recessive allele to have white teeth. So they only need 1 X to have brown, and they only need 1 X to have white. Whereas females have 2 X chromosomes. So if we order them to have white teeth, they need to have 2 recessive alleles. Okay, so they need to have 2, whereas males only need to have the 1. Right, this is just the last thing that I'm going to quickly do is from the diagnostic report. What were the findings? What were the issues that learners had with this? And you can apply this to a lot of the mistakes that learners make with monohybrid crosses. Some candidates had difficulty stating the phenotype of the monohybrid cross. They had to write that it was a male with brown teeth and a female with white teeth, and not just brown teeth were crossed with white teeth. This is specific for sex-linked. Okay, when it comes to sex linked, you must say it's a male and it's a female, and then the trait. Where do you get it? From the information given to you. Some candidates gave their own genetic notations when using the genetic cross. So in the previous example, it was supposed to be like this or like this, but they decided to use N, or they decided to use S, and they did their own thing. White, W, for example. You must use what was given in the question. Candidates gave the phenotypic ratio as 2 females with brown teeth and 2 males with white teeth, instead of stating the smallest ratio, which was 1:1, and that's how they lost their compulsory mark. Learners must use the phenotypic description stated in the question and not use terms such as normal, male or female, and affected. Affected should also be avoided unless it is used in the information. So if they give you information and they say an affected male with hemophilia, if they use those phenotypic descriptions, then you're allowed to use it. But please stick to the phenotypic descriptions that are given to you in the question. Lastly, sex-linked disorders can be carried by a recessive or a dominant allele. Very, very important that it's not always the recessive one. Okay, it's very important to understand that. So if it's a female and the disorder is caused by a recessive allele, then the female will if it's caused by a dominant allele, then the female only needs 1. Okay, and if it's caused by a dominant allele, the male will also need 1. But if it is caused by a recessive allele, then the disorder, then the female also needs, the male is only going to need 1, yes, but the female is going to need to have 2. Can you see that? So if the disorder is carried by a recessive allele, the female will need to have both of those in order to have the disorder. But if it is caused by a dominant allele, the female will only need 1. So it's very important that you read the information and you see, is the sex-linked inheritance a dominant characteristic or a recessive characteristic? Okay, so it's a little bit more complicated to the normal genetic crosses that we did with Sina at the beginning, but you just need to keep your wits about you. You are only going to use X's and Y's and males and females when it is a sex-linked inheritance, not when it comes to any male or female that is being crossed. Okay, this is your homework activity. It's about polydactyly. It's a condition that leads to the extra fingers or toes, and it's caused by a dominant allele. Okay, so they want you to do a genetic cross to show the percentage chance that their children will have polydactyly. Okay, so please remember those little tips that I gave you. How do you get your phenotypes? How do you get your genotypes? And then you can work out the cross. Okay. So the next lesson, it's going to be next week, Thursday. Please join us for the second part of genetics. That's where we're going to be finishing off. We're going to be doing dihybrid crosses. We're going to be doing mutations, genetic engineering, and we are going to finish up this topic of genetics. Okay, so please be there, 27th of March at 2 o'clock. Thank you.